A very good morning, students. Today we are going to understand how differential leukocyte counting will be performed. And to perform this differential leukocyte counting, we need certain materials, and I'm showing you the materials here. We have cotton with ethanol to wipe our fingers to make it sterile. We will be also using glass slides. Apart from that, we'll be using lancet. There are two types of lancet. Of course, I'm going to explain you about that in detail. As of now, I'm just showing you a lancet of a very recent model, which is shown here, which has a lancet plastic cassettes also. Apart from that, we'll be also using regularly a lancet which is made up of stainless steel. Okay, I'm going to show you the lancet which is going to be made up of stainless steel. This is the lancet which is going to be made up of stainless steel, and it is sterile. As long as you do not open the cover, it is sterile. So you can use this lancet, and I will teach you how to use this lancet in a short while. And we will be also requiring the blood smear slides. Okay, so these bloods. So we are going to do differential leukocyte count, and for doing this differential leukocyte count, we need a lancet. We have two types of lancet. One is made up of stainless steel, which is sterile, and we can use either one of it. And the other one is lancet, where we can use this lancet for months, and we can throw this off. So we have a sterile needle which is already been impregnated into this plastic cassette. What we are supposed to do, we just have to take this lancet, insert into this container, and then you have to fix it. And once you fix it, you have to tighten it, and then there are adjustments which are available here. So according to the adjustment, the deeper the lancet will be inserted. Okay, so you can choose of medium level, so that prick is going to be a medium. And we also have a lancet of made up of stainless steel. So this lancet, if you look at it, you can see a blunt end and a sharp end. Okay, so this sharp end is actually used to pierce into the skin, and when you are opening this lancet, you are supposed to open the lancet in such a way that you open at the blunt end. Okay, so when you open at the blunt end, and you have to use it like this, you have to hold the blunt end and then take the lancet out. So once you take the lancet out, you are not supposed to touch this tip part at any cost, because this tip part is sterile. So if you touch it, it will become unsterile, and when you prick it, the contaminated surface, if it contains a microorganism or pathogen, will still enter inside your skin. So in order to avoid that, you are supposed to not touch this tip at any cost until you finish pricking your blood. Okay, and how to prick the blood? You are supposed to use any of the finger, but the comfortable finger will be the index finger here. And what you are supposed to do, you are supposed to make sure that the blood is collected at the tip of the finger. So how to do that? You just squeeze it like this, okay, and when you squeeze it like this, you can see that the blood will be collected in the tip of your finger. Once it is collected, you are just going to do a prick by keeping the lancet here and then tapping the lancet on the head part. Okay, so the head part is here, and you can gently tap the lancet on the head part like this. Okay, and you can see the blood will be coming. Okay, so this blood is going to be taken for today's experimental purpose. So what you are going to do, I am just going to keep this blood, one drop of blood, onto this blood smear slide. Okay, one drop of blood is enough. Okay, so once I keep, I should make sure that my blood should clot, so I apply a cotton here, and after that I am going to use a slide, and this slide has to be kept in a 90-degree angle like this. And the procedure to apply the smear, make a smear, is to keep the slide on the blood spot. Once you keep, it is going to make sure that the blood is going to be spread onto all the corners of the slide by capillary action. And after that, you are supposed to gently move the slide up. Okay, when you do this, move the slide up, it is going to make sure that the blood is going to be collected at the
tip of the slide, and you have to use your thumb here, and then after using a thumb, apply a pressure on the thumb, and your index finger should apply the pressure onto this second slide. And now after ascertaining that the pressure is applied, you have to gently slide it out like this. Okay, so once you do this, we are just going to dry the slide. Okay, so you can see the blood has spread in an even manner. As you can see here, there is no thick and thin area here. The blood is spreaded in an even manner. So once the blood is spreaded in an even manner, you can confirm it by looking at under the light. And once you see that there was an even spreading of the blood is happened, you can keep this slide in a place where the blood has to completely dry. Okay, now I am going to teach you another method of pricking the blood by using a advanced lancet, wherein this we have use and throw lancet cassette, wherein you can see the tip of the lancet is going to be pointed in, and you can, as I told you in the previous explanation, you are not supposed to touch the tip of the lancet. So once you take this out, you are supposed to insert this cassette into this like this, and once you insert it, you can gently put this back and you have to turn it to make it tight. And as you can see here, there are numbers which are shown here. So this number is nothing but the extent or the strength of the lancet to pierce your skin. As you increase the number, more the deeper the lancet will enter. So I am just keeping it at 2, and after I keep it at 2, I have to lift this lancet at the back. Okay, so when I lift it, it is going to apply a pressure according to the number I set. After that, I just have to hold this lancet on the index finger, and my thumb has to be holding the button which is seen here. So when I apply pressure on this button, according to the number, the lancet is going to be released and then immediately being withdrawn. So this fraction amount of time is enough for the lancet to pierce the skin. Okay, I'm just going to show you how to do that. You just have to hold the lancet tip onto your finger firmly, and after that you just have to gently press this button. Okay, once you press this button, the lancet will pierce it and then it will go back. But before that, as I told you, when you want to do a pricking, you are supposed to collect your blood at the tip by applying a pressure from the bottom of the finger, and you can see the blood which is collected in the tip of the finger. And after that, you just have to keep it here like this, and then one small press. That's it. Okay, so the blood is going to come. Now I can use the same blood. Okay, I can put this onto the slide. And I will apply cotton, and the trickiest part is the smear. Okay, so you have to apply a thumb onto this tip of the slide, and it should be keeping at a 90-degree angle, and the pressure has to be applied by the index finger of the right hand onto the slide. Okay, once you apply a pressure, you just keep the slide below the blood and then gently move the slide above. Okay, once you move the slide above, you can see that because of the capillary action, the slide had accommodated all the blood from this corner to that corner. So after you do that, you have to gently move up and then slowly drag it down. Okay, so when you drag it down, this blood is going to be formed in a smear form, and this smear is going to be of a thin smear. Now after 5 minutes of time, the blood is completely dried. Okay, so you should make sure that the blood should be completely dried. Okay, if it is not drying, then when you apply the staining solution contains the fixative. The fixative will not fix the cells properly. So you have to make sure the blood that you made a streak has to be completely dried. And once it is dried, I am just going to keep it in the drying stand. Okay, like this. And I am going to apply the Leishman stain. Okay, we had prepared the Leishman stain, and just take this Leishman stain, apply it onto the slide like this. Okay, so you should make sure that the slide should be entirely covered with this Leishman stain. all the part of the slide has to be covered with this stain. So we'll wait for 2 more minutes, and after that I'm just going to discard the stain and I'm going to wash this stain. So 2 minutes of incubation is over. So what I'm going to do, I'm just going to gently wash this slide by applying distilled water at the top. Okay, gently I have to apply so that I wash the stain but not the sample that was been spreaded as a smear. Okay, so gentle wash is required. I just have to pour the water in such a way that it falls onto the sample very slowly. At the same time, it removes the stain. The stain removing means the place where the sample, the blood sample is not there, at that particular place, if you add water, the stain will be removed off completely. Okay, so this is how you're supposed to wash your slide. Okay, and there are few stains at the back. I can also apply more pressure at the back because there is no sample present on the back. So I am just applying water on the back to remove the stain, and 1 to 2 minutes of washing is required. Okay, so after this wash, what I am supposed to do, I am supposed to completely dry this slide, okay, under a fan, okay, in order to make sure that the water are going to be completely dried off. So for that purpose, I am just keeping it in the same stand where I added the stain just for drying purpose. So now it is going to be drying for another 5 to 10 minutes, and once it is dried, I am just going to take the slide and then I am going to observe it under the microscope. Okay, I am going to teach you how to use the binocular microscope for today's experimental purpose. I think that you already know how to use this microscope in previous labs, but this is a binocular microscope wherein there are 2 adjustments are there. You can see a big ring which can be moved forward and backward, and a small one which can be also moved forward and backward. So this is going to be a coarse adjustment and this is going to be a fine adjustment. So if you do a coarse adjustment, this slab is going to move towards the lens if you do a forward movement, and if you move a backward movement, this slab is going to go down. And the place where you are going to keep the sample is going to be here. So you are going to keep the slide, okay, here, and we have a slide rack available in this microscope. Once you keep it, you are supposed to keep the rack in such a way that the light has to be absorbed by the sample, and the light will be transmitted through the sample. So you have to move the slide in such a way that your lens and your specimen and the light should point on the same direction. Okay, and you can adjust this lens. There are 4 lenses are available, starting from 4x, 10x, 40x and 100x. 100x is going to be oil immersion. So for today's experiment we will be using 4x, 10x and 40x. 4x is used to focus where the samples are, and 10x is used to look at the samples in a little detailed manner, and 40x will show you the nuclear morphology. With the help of this nuclear morphology, we are going to identify what cells are going to be the WBC based on the nuclear morphology. Okay, so after the water wash, the slide is completely dried. As you can see, the smear is very even, so that the very thin smear is present from top to the bottom, but actually you will be having a head part, body part and tail part. Okay, so tail part will be of very thin smear, head part is going to be of a little thick smear, and in the body part we will be getting an appropriate amount of smear that can be visualized with the microscope. So head part, body part, tail part, and you are supposed to focus in the body part as well as in the tail part. So before you focus, you are supposed to write down the different types of cells: neutrophil, lymphocyte, monocyte, eosinophil and basophil, and while observing it under the microscope, you are just going to mark down how many neutrophils that you come across. So again, based on the morphology, we are going to know whether these cells are neutrophil, lymphocyte, monocyte, eosinophil or basophil, which the faculties is going to give you an explanation about the morphology and other things in the lecture session. But in the video session, I am just going to show how to count the cells using this microscope. So now I am going to place the slide in the slide slab, and then I am just going to lock it, and after that I am just going to turn on this microscope, and after I turn it on, as you can see, the specimen is present between the light and the lens, as you can see. And I'm just going to move this lens slab, slide slab little bit forward, backward, right side, left side. Okay, so according to the thin of the section, you can move it on either direction. And first point that you are supposed to do is you have to change your lens in such a way that the lens is fixed at the very low magnification. Okay, and once you fix it at very low magnification, you are supposed to lift the slide slab, the specimen slab, to its maximum position, and after that it is not going to move even if I do the course adjustment. And from now onwards I am supposed to observe the specimen. Okay, I can use this two eyepiece of the microscope to observe the specimen, and once I observe this specimen, I can see the RBCs. There are more of RBCs, and once I see this specimen, I am just going to show you.
Okay, now we are looking the cells at 4x magnification, and this is how it will look when you look at 4x magnification. And when you look this specimen, you can see that there are spherical shaped structure cells are there, which are literally RBCs. So most of the cells are going to be RBCs, and there are very few cells which are looking blue in color, which are stained blue. Those cells are WBCs. So we will be seeing a picture something like this at 4x magnification. Now I had changed the lens to 40x magnification, and as you can see, we had magnified the RBCs, and as I told you, more of RBC cells will be there. And again, if you look at very carefully here, you still tend to see the cells which are looking blue in color, which are the cells of WBCs which had taken up this Leishman stain, and that is the reason why it is looking blue in color. And in fact, if you can able to closely look at it, you can tend to see the nuclear morphology. And the morphology which is shown here is an example for a lymphocyte, which is shown at the bottom, okay, on towards my left hand side is lymphocyte, because it contains full of nucleus which is completely stained blue in color. And the cell which is shown in the center seems to be neutrophil, which is multi-lobed cell. Okay, and the cells on top of it is again is going to be of neutrophil. So we can see two neutrophil and one lymphocyte at this 40x magnification. Okay, now I am going to I am showing you the animated file of the WBCs, different types of white blood cells. And the very tricky part about today's experiment is you have to identify the nuclear morphology. Based on that, you are going to designate that particular cell as a neutrophil, basophil, eosinophil, monocyte or lymphocyte. So if you look at the images, you can see that the lymphocyte, okay, the cell's size is little small, and if you look at the size of the cell, the entire cell, almost entire cell is going to be occupied with the nucleus. So that is why in the previous picture of 40x, I told you one lymphocyte is seen at the bottom left hand side, right? So these cells will be completely filled with blue in color. So if you can see similar morphology, then you can designate that cell as a lymphocyte. And the monocyte will look like a kidney bean nucleus. Okay, if you look at the structure of the monocyte, it is not going to occupy, the nucleus is not going to occupy the entire cell, but in the center you will be looking like a kidney bean like structure nucleus. So if you can able to identify that, then you can designate that as monocyte. And in the case of eosinophil and basophil, both will look bilobe. Bilobe means two lobes, two distinct lobes will be seen, as you can see in both the eosinophil and basophil. Then how can I differentiate between eosinophil and a basophil? So if you look at the eosinophil, the lobes are joined to a very thin fragment. Okay, whereas in the case of basophil, it will literally look like a headphone that we used to use. Okay, so if you look at a structure, a nuclear structure which has a headphone-like structure, then you can designate those cells as basophil. But if it is bilobed and these two lobes are just interconnected with one another with a thin strands of a structure, then it is called as eosinophil. In contrast, if you look at neutrophil, you tend to see there will be multi lobes. Okay, so it starts with 3 lobes and it can go up to 5, 6 lobes also. So these cells are called as polymorph. Okay, polymorph is it is going to have different morphology. So neutrophil will be having multi lobes, more than 3 lobes. If you can see, then you can designate that cell as a neutrophil. Now let us look at the actual slide and how those cells are going to look like. So I'll be going from lower magnification to a higher magnification, and we'll just observe that.
After you focus the specimen and low magnification, I am just going to change this 4x lens to 10x lens just by tilting it like this. And once I tilt it, I do not have to change the focus. Okay, I can immediately see this specimen, and by looking at this specimen, I am just going to do the fine adjustment just to accommodate to my vision sight, and then I am just going to slowly scroll down this slide slab. Okay, so such a way that I can able to see the cells which are going to be stained with a blue in color, because this Leishman stain is going to give you the stain, and I had currently focused a cell which I am going to show you. As I told you, there are a lot of RBCs are going to be there, and in center of the RBC I am just going to focus a cell which I am going to identify it as lymphocyte, because it is going to be having a full nucleus and the entire nucleus is been stained with the stain. I am just going to show you that.
Okay, so you can see 2 lymphocytes which are sitting on the right and top region, wherein both the cells are fully occupied with the blue color stain, and those 2 cells are a good example to understand how a neutrophil will look like. And in the forthcoming slides, I am just going to show you an image which is already marked with the different types of cells. So once you observe, you can able to completely understand how these cells will be looking like, and so that you can able to visualize, and you can able to designate the cells appropriately according to the nuclear morphology.
So we had counted the cells in such a way, when I am looking at under the microscope, I have to count and then note down how many neutrophils are there. For in today's experimental session, I had found that 1, 2, 3, 4. 4 neutrophils were counted, and 2 lymphocytes, and 2 monocytes, and 1 eosinophil, and I couldn't able to find basophil. Sometimes you might find, sometimes you might not find. So accordingly, I had noted it down as neutrophil as 4, lymphocyte as 2, monocyte as 2, eosinophil as 1, and basophil as 0. Okay, to calculate the different percentage of the cells, the formula for calculating is number of cells that you are going to calculate. So in case if I am going to calculate the percentage of neutrophil, the neutrophil number is going to be 4 divided by total number of cells. So total number of cells is the cell, different types of cells, how much ever you counted, that is going to become as a total. So in this case 2, 4 plus 4, 8, and plus 1, 9. Okay, so 9 is the total cells. So for calculation, I have to include 4 neutrophil divided by 9 into 100 is going to give you 44.4 percentage of neutrophil. Similarly for lymphocyte, 2 cells I counted, so 2 divided by 9 into 100 is going to give you 22.2 percentage of lymphocyte. And monocyte also the same number, so you have to calculate 2 divided by 9 into 100 is going to be 22.2% monocyte. And for eosinophil, 1 is the eosinophil count divided by 9 into 100 is going to give you 11.1 percentage eosinophil. And in today's experiment I could not find basophil, so it is going to be 0. So 0 divided by 9 into 100 is going to be 0 percentage of basophil. So these results are supposed to be compared with the normal values. Okay, so the normal values is for neutrophil, it is going to be of 40 to 75 percentage, so which falls in the normal category. And when you take the lymphocyte population should be between 20 to 50, and again my calculation is also falling under the same category. And for eosinophil, it should be falling between 1 to 6 percentage of category, but in today's experiment I got 11%, which means the percentage of eosinophil is increased. So that means I might have allergic responses. And in the case of basophil, it is going to be 0%, and the normal value for basophil is going to be between 0 to 1%, which is also falling under the same category. So out of this today's experiment, I found that increase in the eosinophil number. So I have to, in the discussion part, I have to write a discussion what could be the probable reason wherein eosinophil numbers goes up. So that has to be written in your discussion part, and the result part has to be written in such a way that what is the percentage of neutrophil, what is the percentage of lymphocyte, monocyte, eosinophil and basophil.